Hi, my name is Pamela Akuku, and today I'm going to tell you about what I actually do. I know a lot of people just know I'm in the basement all the time with bones, but we'll see what I actually do with those bones. So this is the first site that the project that I'm part of, and Maria Soto as well is part of, we ever worked on, and the site is called Ewass Oldupa, which is Masai in Masai language, and it means on the way to the gorge. So, hi, welcome back. So the project is composed of like various universities, various individuals, and everyone has their own skill set. So I'm going to do like a short 2-minute background, which Maria and I did a couple of years ago, but I know some of you were not here, so I'm just going to do that. And then we'll just talk everything bones, okay? Okay. So the site, Ewass Oldupa, is located here on the western gorge of Oldowan. And we have here like an extraction of it, and you see Ewass Oldupa and some of the most famous sites in Olduvai Gorge. Epilke Zinj, because you have all heard of it. Sinthropus was found there. There have been anthropogenic marks and whatnot. And then DK right here, because it was formerly the oldest Oldowan site, but it was found on the Eastern Gorge until we came along. So in 2018, uh, 2018, the project got permission to kind of survey all of these sites, and with the agreement that they could excavate 3. So during the reconnaissance mission, of which Maria Soto was part of, they found this eroding from what was then known as geolocality 63, aka Ewass Oldupa. So for this reason, they decided to exploit, but not only for this reason. Another interesting fact is that it is only located 300 meters away from geolocality 64, which is super famous because that's where Olduvai hominin number 65, here, a maxilla of Homo habilis, which we actually got to see the actual one with Raquel last year at Arusha National Museum, was discovered. So for this this reason, we decided to explore Ewass Oldupa. And the pilot season, the pilot study was in 2018, where they did like test pits. And then we thoroughly excavated it in 2019, 2022 for 4 months, and 2023 for 3-4 months. Okay, fun stuff. This is the trench that was done, the test pit, right? And I want to draw your attention to this right here. It is a tuff, right? So when Maria et al. Were excavating, they found like stone tools and bones eroding from under here. And they were under the impression that this tuff they were seeing right here is tuff 1B. Which is cool because Stuff 1B is known to be 1.8 million years old. So they were working under the impression that, oh, everything under here that we are seeing is slightly older than 1.8 million years old, right? But then they actually found out that this is not Stuff 1B, but actually it's Stuff 1A, which is 2 million years old. So everything that they were seeing under here is older than 2 million years old, and hence why Ewass Oldupa is currently the site with the earliest Oldowan material, because the previous one is 1.848, right? DK, right? So we did a strat section to kind of show you what we did. So we made a total of 6 trenches, more or less. So the tuff that I was talking about, tuff 1B, is right here. This is the 1.8. This is what we thought we had. But what we actually had was this, tuff 1A. And so all our trenches are below tuff 1A, all the way to just like above the Nabi Igneum Bright, which is a little older than 2 million years old. So these three trenches, trench 2, trench 3, trench 5, are where all of the faunal materials came from. Trench 7, right here, our oldest, biggest trench, nicknamed Australia, did not produce any fauna. But we did find lithics from here, making them like the oldest Oldowan now from Oldowan. Okay, fun stuff. So fauna, the fauna that we found, the methods, we looked at everything greater than 2 centimeters. We classified it into ID and non-ID, and then the modifications were studied under a microscope. And then further modifications, like interesting ones, we used the Hyrax microscope. The total number of fauna recorded was 3,661. And with an NISP means like number of identifiable specimens, was 1,157. There were 565 lithic artifacts discovered. I'm not gonna talk about that. Fauna only today, sorry. So, and then we identified all the specimens down to taxa, and then we use the dental remains specifically from bovid because we can get them down to tribe level to create what we call a ternary plot. You will see later. It's so beautiful. Okay, so a total of 22 taxa were recorded. And as with most African Pleistocene sites, the bovids were the most common, like the most dominant taxa. But we have also other things recorded. We have like equids, suids, and then we also found primates. And there's a funny story about these primates I will tell you. There were other water-dependent species as well, such as like your crocodiles, your turtles, and also hippo present as well from the taxa we have. This is just like a table showing you like the NISP and MNE per trench from the 3 trenches that we found the fauna from. And then I have some pictures, examples of some of the fauna that we found. These are the crocodile teeth. And this is like a plastron of a turtle. And in the corner here, you see a tooth mark, right? A puncture. There's a bit missing. We found a bit of an elephant tooth, and then this is the beautiful primate that we found. Now, it was interesting because those of you that deal with like hominins, if you look at this radius, the morphology is almost the same. If you didn't know any better, you just look and be like, yes, we found a hominin. And that was what we thought. And we consulted like various hominin specialists, and they were like, oh my God, congratulations, you actually found a hominin. And then we found a student who deals specifically in the radial bone of a Therapithecus oswaldi, which is a type of primates. And this is actually what we have, and not a hominin, as we thought before. But still cool. Still cool because on the left-hand side of the screen, this is a recreation of Therapithecus oswaldi that they found in the Okote member of Kubifora in Turkana. And the Okote member is 1.6 million years old. And our Therapithecus is 2 million years old. Like, that's all I'm saying. I'm not flexing or anything. That's all I'm saying. And then with the animals we found, it was really, really interesting because so we kind of lumped the group. So, you know, you have like from your size 1 to size 6 in African taxa, and size 6 being elephants. They're the largest. So what we did is, so class size 1 and 2, so from about like 6 to 84 kilograms, we say that they were like small, medium animals, which is the SMA. And then the MMA is size 3 and 4, and then the LMA is 5 and 6. And then we had others that were unknown because we have really fragmentary pieces. Most of you have seen me study these fragmentary pieces and be like, is it bone? Is it stone? I promise, it's bone. They're just fragmentary. But our largest or most represented size class was the medium size class in all 3 trenches. And this is interesting because, okay, we'll get that to later. So we also did what we call a skeletal profile. We did it 2 ways. This is NISP. It's normed number of identifiable specimens. So when you're doing your analysis, usually you'll find, like, let's say, for example, femurs, humeri, and all that, and you don't want to overcount your specimens, so you will norm them. That means that you will divide it by the number of times it occurs in the human body. So if you have humerus, you have 2 humeri, you will divide it by 2 to norm your specimens. So that's what we did here to see what is represented more. Like, is it the skull elements, the forelimbs, the hind limbs, the distal limbs? So when we put it, like when we total it, when we put all of them together, regardless of whether it's small, medium, or size, we see that, like, with the hind limbs and forelimbs, there's more or less an equal representation, and the distal don't fall far behind, but we don't have a lot of skull elements. But if you look at, like, the medium size class, it's more or less like the same. The number is like, so what this means, like, there's not that much of a disparity in the skeletal elements. So what you're looking at is not a biased assemblage. And then this is what we did with the bovid teeth. We created what we call a ternary plot. So the first premise of a ternary plot is to see, like, what composes your assemblage predominantly. Are they young? Are they prime? Are they old? So if your elements fall here, then it's predominantly a prime assemblage. If it falls here, it's a young individuals is what I mean. And if it's here, it's old individuals. So this the stars show you the trenches that they came from. So the trench 2 ones, the trench 3, and trench 5. So all of our specimens are predominantly prime. They're like their elements in their prime. And then we compare them to, like, animals from actualistic studies from Stinner and Bunn and Pickering that have been hunted down by carnivores to see, like, where they fall on the ternary plots, to see if they overlap, and they don't. So what does this mean? We don't know. Kidding, we do know. I'll tell you what it means. There's also, like, a bunch of modifications. We found 2 cut marks, which was really cool because, like, in most open air Pleistocene sites, it's really, really difficult to find and identify and just, like, ascertain these cut marks. We also have some percussion marks present, but I would like to draw your attention to, like, the abrasion and the rounding. So we have, like, this, what the highest, the highest modification, the most dominant modification. But this is common in open air assemblages, and especially, like, with Oldowan Gorge, where you have like the environment, the paleo environment we were looking at, there was like a fluvial environment. There's flowing water. So as the bones are being moved by water and also by sand, it causes, like, rounding and also abrasion and sheen. So some of them are very brilliant, like you think I applied, like, nail lacquer on them, but these are some of the cut marks. And this picture is not very clear, so I'm going to show you, like, one by one. I think in the next one it will be better. Can you see? Is this okay? Is it better? See? Okay, so this is like a long bone fragment. And this was very interesting because when I was looking at it first under the microscope, I wasn't sure because, as you can see, there's 4 different striations. There's this, there's this one, and there's this one, right? So we had to use like the Herox microscope to kind of just like be sure. And at the end, we were sure like these two are cut marks, but these two are scores, right? And it becomes difficult, but this is something that is very common in open air or even Pleistocene sites. There's different access to resources. Both carnivores and hominins are competing on the landscape. And as you can remember, like if you read literature in the 80s, 90s, there was a big, big debate on scavenging versus hunting, fast access. They came up with like models like, oh, carnivore to human to other people, or hominin to carnivore to. So this is part of the problem. Why? Because as you can see, this is a long bone. But even as you look at it, are there cut marks? Yes. So that shows that the hominins are active and they're doing things on the landscape. But are there like carnivore scores as well? Yes. So carnivores are active and they're doing things on the landscape as well. But none of them is overlaid on top of the other. So you can't tell who came first, right? It's like the whole question of the chicken or the egg, which came first? You don't know. But in the second one, this is a rib, and this is like a very distinct cut. So this was cool because this is on the ventral side of the rib and not the dorsal side. So the ventral is like the inner parts of the rib. And this is usually used to indicate fast access because like imagine you're dismembering or like defleshing your carcass. So you have to like open the rib cage and you're removing things. So if you're coming second, why would you need to deflesh the ventral side? There would be barely nothing left, right? So this kind of indicated that hominins also have fast access to the the resources that we have here, right? And then percussion marks we found. So we found different types of percussion marks. These are notches, and they are a good indication of percussion marks. There were different types of notches, but I chose this 4 because usually incomplete notches are taken as an indication of hominins. When carnivores bite down on a bone, like with their canines, usually the notch will be complete. They will bite right through the bone. But if you can see, doesn't this look kind of similar to when you're flaking your stone tools and then when you get the flake scar that is left behind? Same during bone breakage. So these are the anthropogenic percussion marks that we found. So yes, hominins are active on the landscape, but also carnivores are active on the landscape, as you can see from this very beautiful puncture mark presence, and also like the furrowing on this humerus. And none of these elements have any form of cut marks on them. So in this case, primary access to the carnivores, but in the previous case, primary access to the hominins, right? So we have kind of like a mixed scenario going on here. We did box plot analysis because of the tooth marks that we were finding, and our results indicate, so this and this is what is representing our sample, the first one. And then we compared it to other actualistic studies. And from that, you can see that we can say it's medium to large size carnivores causing the damage or like having access because of where they fall. They fall like ranging in like the hyena range and not in the smaller carnivore range. So there's medium to large size carnivores active on the landscape, but the hominins are also still active on the landscape. And then we also looked at bone breakage patterns because as we found the percussion marks, it means like they're kind of breaking the bones, maybe for marrow access. And our most predominant one was smooth and oblique angles, which has nothing to do with anthropogenic modifications of carnivore. It is natural modifications, but it's an open air site. So maybe it occurred after the bone breakage. They modified. I'm going to show you an example with pictures here. So these are some of the bones that we find But I wanted to show you because I think this is interesting. This is a phalange, right? And I don't know how many of you have worked at Abrik Romani, but as I was looking at this, Palmi said that she also finds this a lot at Abrik Romani, where there's predominantly like Neanderthal activity, and they find that the phalanges are broken this way, but we don't have any explanation for why, because if you know, like there's barely any marrow in the phalanges, so then why would you break them like this? And for what purpose? We don't know. We'll figure it out. We also did like a kernel density test to kind of see what is the relationship between the accumulations, like, because we are finding lithics, we are finding fauna, we are finding percussion marks, we are finding cut marks. Can we find a relationship between these two? So we wanted to see that. And the answer is sadly, no, we cannot find we cannot find a direct relationship. But again, this is very common, even in sites like FLK Zinj and DK, they have the same problem. So what does this mean? We'll start first with the taxa. So because we had a variety of taxa, it shows, and maybe you read this word a lot, a mosaic of environments. So we just have mixed environments. There's different things going on. We have open landscapes, we have woodlands, bushlands, and we also have water sources nearby. And we can just infer this from the number of taxa and variety of taxa we are finding. So anthropogenic modifications are indeed present at the oldest Oldowan, which is good. We are adding more to like what we know about Pleistocene hominins. But the assemblage may be a product of independent accumulative processes. And this is because we could not find a direct correlation using the density, the kernel density, and also other statistical methods that we use. Like we cannot find direct evidence that yes, they use these tools to process these animals, and beautiful story, this is what was happening. So they're there, the carnivores are there, but what exactly is happening? We don't. So like I said, like we are part, the project has people with different skill sets. And so when I said like a mosaic of environments, I use the taxa, but there's people who study phytoliths, and this is what they found. They found that there's C3 and C4 dominates, meaning that there's like mixed plants in the area. There's mixed environments. So basically their results just supported what I'm saying, that there is a variety of environments and habitats in the same place. Why is this important? Why do I keep saying this? Well, because if there is a variety of environments, then it means that there is some sort of like behavioral complexity and flexibility in the hominins, because if the environment is constantly changing, there's different things, there's medium to large size carnivores, but they're still having primary access to medium sized animals like class 3, size 3 and 4, like horse size going upwards, then they do have some sort of complexity going on, and they have the ability to be flexible with these constantly changing environments. And this might be what facilitated the dispersal of now like the genus Homo to other continents outside, the movement outside of Africa. So yes, so these older groups have the ability to exploit these diverse environments and biomes at 2 million years old, which is really great if you think about it. With the limited technology that they have, it's a great thing. And that the earliest Oldowan assemblage from Olduvai, it provided diverse fauna. And then from this, there's something about Ewass. So Ewass is not only bed 1, but like early bed 1. So basically what we did is we extended what is known of the paleo environment from Olduvai Gorge from earlier bed 1, like what's happening before bed 1 and then to bed 1. So environments have been constantly changing since before bed 1 time. So this is good because we are adding to the information. And these are some of the things we found from the trenches. So this is an elephant scapula. Maria will tell you she had to carry this all the way back. And indeed, it's the size of that guy. It was as big as him. So there's really cool stuff. And these are the wonderful, wonderful people that make our work really easier. And actually now, finally, actually, like, thank you. If you have any questions, please.